antimicrobial effects of anthocyanins and polyphenols from low-bush blueberries on E. coli, Esenterica, and S. aureus. Polyphenols are secondary metabolites of plants to protect from ultraviolet radiations, fungi, and bacteria. Polyphenols are antioxidants, anti-inflammatories, and contain antimicrobial properties. Anthocyanins are a type of polyphenol that are responsible for the red to blue pigment on flowers and fruits. Anthocyanins have many health benefits, especially preventing diseases brought on by oxidative stress. With the rise of antibiotic resistance, there's been an interest in polyphenols to limit the spread of foodborne pathogens without using chemicals. Most of these studies use polyphenols extracted from low-bush blueberries due to their high concentration of phenolic compounds due to the acidic environment they grow in. Anthocyanins are thought to be the reason for the health benefits of polyphenols. However, there have been fewer studies on anthocyanins as their stability is heavily affected by temperature, pH, and light. Polyphenols have been shown to damage the outer membrane of E. coli, Esenterica, and S. aureus. It is hypothesized that anthocyanins extracted from low-bush blueberries will inhibit the growth of E. coli, Esenterica, and S. aureus by affecting the outer membrane. The objectives of this study is to extract polyphenols and anthocyanins from low-bush blueberries, determine their antimicrobial effects, and investigate if the treatment of anthocyanins and polyphenols affect the cell wall proteins in E. coli. Polyphenols and anthocyanins extracted from blueberries were quantified using an F&C assay and pH differential. Anthocyanins were identified using UPLC. The bacterial inhibition was investigated through MIC assays and COI plates. The relative gene expression of HDEA and CFA were looked at using PCR. Delphinidin-free rutinoside, cyanide-free glucose, and melvidin-free glucoside were present in the anthocyanin extract. In the polyphenol extract, there was 1.49 milligrams anthocyanins per gram of blueberry and 0.46 milligrams anthocyanins per gram of blueberry in the anthocyanin extract. In the polyphenol extract, there were 232.13 micrograms per ml gallic acid equivalent per gram of blueberry of phenolic compounds. In the anthocyanin extract, there were 60.65 micrograms per ml gallic acid equivalent per gram of blueberry. The COI showed that the neat polyphenol extract and anthocyanin extract inhibited the growth of E. coli, Esenterica, and S. aureus. The zone of inhibition was significantly different from the vehicle control of 0.1% TFA and TI water. The minimum inhibitory concentration of anthocyanins and polyphenol extract for E. coli was 3.125% concentration. Esenterica and S. aureus had a minimum inhibitory concentration of 3.125% polyphenol extract and 6.25% concentration of anthocyanin extract. The PCR showed that there's no significant difference in the relative gene expression of HDEA and CFA when compared to the controls. In conclusion, this study found that anthocyanins and polyphenols were successfully extracted from low-bush blueberries and inhibit the growth of E. coli, Esenterica, and S. aureus. There is no significant difference between polyphenols and anthocyanins in the effectiveness of inhibiting bacterial growth. The treatment of polyphenols and anthocyanins had no significant change on the relative gene expression of CFA and HDEA. The hypothesis that anthocyanins extracted from blueberries inhibit the growth of E. coli, S. enterica, and S. aureus by affecting the outer membrane is rejected.